And okay, so in the Congress here, I'm not exactly a person. Okay, I'm not a mathematician. I am working in structural biology and more in bioinformatics method for structural biology. And I'm working in Nancy in a laboratoire called Laboratoire de Physique and Chimie Théorique. And actually, I'm going to show you some method we are developing using the distance geometry approach, approaches coming from Antonio and co-worker of Antonio for studying the conformational space of intrinsically disordered protein. So actually, I'm talking to the following problem, which is the molecular distance geometry problem, which was formalized by Leo Liberty, Antonio Micurino, Carly Lavor. Okay, that's a bit a problem, yeah. But actually, it was also studied by Crippen and Havel in the 90s, and it's a problem which is the problem of finding the coordinate of conformation, which is corresponding to the set of atomic coordinate for a molecule, such that all constraints are satisfied. And actually, the protein I'm going to show later some structure. They can be considered as a geometrical object defined by their atomic coordinate, and we are then studying this object in the computer. So they are in silico protein. And okay, so the structural biology is a field which is developing since some decades, and when we have something which is called a folded protein, the structural biology is based on the fact that we are trying to obtain the convergence of the protein. For example, here we have a protein is a polymer of amino acids, so it's a linear chain of amino acid, but using NMR, so nuclear magnetic resonance measurement, it's possible to fold it into a structure, and we are doing the calculation several times, and we are very happy if all the structures are superimposed actually, and then we are getting this here you see that the yellow part is very well superimposed and the green part is a bit more blurry, and then you have been converging and you are happy. For example, if you are doing something which is very popular during the last year, which is the cryo-electronic microscopy, you are having some images, and then you are trying to get convergence of the images, and then finally you have some particle, you get some converge structure of the particle, and if you are sufficiently lucky, you even get structure at atomic resolution, so you even see the atoms. And all that is based on the convergence of your signal. And okay, so you have another case in which you have a part of the structure you can superimpose when you are doing the ensemble. You are calculating an ensemble of conformation, but other part is very disordered. So you have a part in gray here which is well superimposed to each other, and other part which are going in all direction, and that's the case of disordered region, or the so-called also intrinsically disordered protein, which are proteins which don't have a defined conformational structure and which is converged. And this intrinsically disordered protein, they are playing a very important role into many biological processes. Related, for example, to signaling or to all kind, or also to several disease. And so in this frame, we cannot apply a method for which we are searching for the convergence of coordinate. So, and it is in this frame that then the method developed by Antonio and the co-worker were becoming interesting. So the method is based on the fact that we are positioning the atoms given a certain order, and the order is chosen in the way that we are getting a click actually. So that we are getting, when you have positioning I-3, I-2, I-1 atom, you are positioning the atom I, and you know all the distance between the four atoms, and all the green atom green distance are known exactly, and the magenta one can be exact or can be an interval. And if you are doing that, then you can define two positions for I, because actually with this distance you get a cosine of an angle, but you don't get the sine of the angle, so you have two positions. And this method can be also described a bit like can be described as the intersection of sphere. So you have 4 atoms and you want to position the third. You have 3 atoms and you want to position fourth one. So with the each of the 3 atoms C and HN and Ca, which are there, you can define a sphere around it, and the 2 spheres here, C and HN, they are intersecting. They are giving you a circle, and with the last sphere in green, you are getting 2 points in the same way that you had in this tree. You had 2 points, two branches. So that's called the branching step of the algorithm. And then if you have all exact atoms, you get exactly 2 points. If you have one interval, as I show here, you have one interval in magenta, you get 2 circle arcs. And actually you can describe that as a construction of a tree, and you are branching your tree, and then so each time you have positioned an atom, you can check whether you have other restraints which are maybe not satisfied, and then if they are not satisfied, you can prune the tree. And in principle, okay, it could work, but it's a problem which is NP-hard, as mentioned Antonio, which is okay for in another context, but it's also the similar families of problems. And actually, another way to tell it is that you have an exponential complexity, and so you are dead. And so then I wanted to apply this kind of approach to the proteins, and I came out with some kind of method which was a bit unmade method. So the idea is that you are taking your protein and then you are cutting it in segments, in fragments, and then fragments are smaller, so you can apply the construction of the tree and get all the leaves, which are all the solution. So you have all the trees which are in green here. And then what you are doing, what is saving you also, you are doing a clustering. So I was using a self-organizing map for making unsupervised clustering of the conformation in order to reduce their number. And then you are taking the representative clustered conformation for each fragment, and you are assembling them to get the whole protein conformation. So we have been to test the method. We have been applying it on cases of small folded protein for which we knew the solution, and we have been, so here in green that was the initial structure, and in red that's the structure which is the conformation we have been able to produce with this method that we call TAIBP. And we have some decent score, which Rosetta, which is a program which is known for protein folding generation. Okay, so then now what to do with the IDP? For the IDP, something you can measure by nuclear magnetic resonance are thing which is called the chemical shift, because you are using nuclear magnetic resonance, you are putting your protein in a magnetic field, and you are looking to all the magnetic moment of each proton, a bit like a small magnet in the big magnetic field, and they are all interacting, and especially each proton have a resonance frequency, because this magnet can turn around in certain space, and the frequency is related to the electronic environment, average environment of the protein. And so it was very early remarked that when you have so you have two kinds of big different structure you can have in protein. Some are helices, which are represented here, and some are beta strand, and you have opposite behavior of the chemical shift depending when you have a strand and or the alpha helices. And so using that, actually, there have been many methods developed, in particular neural network method, and which is one which is called Talos, we will see a bit later, and another one which is more Bayesian one, delta 2D, I will present also later. So we have, in principle, a method to get some information on the local conformation of the protein, but it's not very precise information at all. It's very fuzzy. Okay, so now I'm introducing the IDP for which I'm going to show some result today, which is the SERV1A, which is studied by our co-workers in Taiwan at Academia Sinica. And it's a protein which is quite, with residue which are very much charged, so either positively charged, either negatively charged, and this protein is involved interacting with other protein. One is alpha-synuclein, and is preventing the fibrillation of the alpha-synuclein. Fibrillation is related to all amyloid disease, neurodegenerative disease. So that's important to know whether we could interact with this protein, maybe finding some ligand interacting with it and preventing it to induce the fibrillation of the other protein. So, okay, so I said we are going to work with the dihedral angle, so I use two programs, so you let me know when I have still 5 minutes. So this protein was studied at 2 pH value, and we were looking at using the two programs, the Talos and the delta 2D I mentioned. So these two programs, so the Talos is providing a probability. So here I'm showing you, which is something which is very used in the social biology, which is called the Ramachandran diagram, which is actually depending of two angles, which are phi and psi, and Wagner Daroche a bit later he will show you a bit more about this angle. So you can plot one along the protein structure. You can plot all the value of the phi with respect to the value of the psi, and you get a map. And when we are doing Talos prediction, for each residue you get some probability distribution for the different, which are giving you some peak. So here you have three regions which are very probable, relatively probable, for one given residue in the polymers. And delta 2D is even giving you a larger region for the prediction, which are larger than for Talos. So as you see, actually you have very imprecise information, not converged at all, because if you are going from one, we could have a possibility to be in that region, and in that region it means that you have a lot of different alternatives for your local conformation. Okay, so for the Talos probability map, the situation is relatively easy. One takes all the possible the maxima of probability, and one is drawing a box around it, and then we will do the constructing the tree by varying the point in this box, actually, alternatively along the calculation. And for when you have the delta 2D, you can see the regions are much bigger, and so we decided to use a more detailed description of the Ramachandran map, which was proposed by the group of Karplus in 2011. And actually we used we were looking for in the PDB in the PDB data set, we were looking for triplets of all this region. So here you have the AAA, which is actually the red region, which is here, and the BBB, which is blue, but it is here, and PPP is this one, which is here. I'm sorry, the color are not corresponding. And then but then that's giving you an idea of the distribution in Ramachandran map for the folded protein. Or we are also looking to an intrinsically disordered protein, and then so we have been using I have been generating I've been taking just protein with 2 amino acid and generating all possible conformation by generating the tree I have been just showing with that was the IBP, the interval branch and prune approach. And then if you look at the distribution of all possible assigned So I was doing that by assigning all possible different regions alternatively for the tree residue, and then you see that for some case we have a lot of solution, and for other case we have not so many solution. And then I was constructing a database of all possible triplet of secondary structure, taking the most populated solution, which are from these two calculations with IBP, plus the folded structure assignment for the secondary structure. And using that, then it's possible to calculate, so to predict. So it is in magenta here, to predict boxes for starting from the delta 2D prediction. So then at the beginning we had a very big region for the delta 2D prediction, but here we are ending up with much smaller region which are in magenta, and I have been superimposing here in magenta it is the delta 2D prediction, and in black you have the Talos prediction. You see that they are recovering a bit, but the magenta prediction are also predicting a lot this region, which is less predicted by Talos because they are less populated in folded protein. So we are using two tree scheme for calculation, either the Talos input, so two input from the Talos with a different threshold for defining the box, and delta 2D input, and then we are generating conformation. So we are generating so here you have some example that we have been generating some conformation, so it's more statistic for the generation, and that's the generation of the assembly. So each representative conformation we are assembling together to get the full structure. So I'm not going to talk to give a lot of detail on it. Okay. Now I just want to show you, to compare that to another method which is called fleximecano, which is generating conformation more on the Monte Carlo approach. And if you look at the distribution of atomic coordinate, you see that fleximecano and TIBP, they have similar distribution. TIBP slightly bit more converged, and also for the what's so-called the radius of gyration, which is giving you a bit an idea of the shape of the ensemble. They are relatively similar. If one is looking at the something I'm calling the local the profile of local gyration radius, which is actually I'm taking some segment along the protein sequence, and on each segment I'm taking a local, so which is the blue, the light blue segment. I'm calculating the gyration radius on it. I can plot this value along the sequence, and I'm getting especially for the Talos structure, the IBP structure, Talos structure, I'm getting something which is a bit with two steps, which is so you have higher value here and smaller here, because here you have an alpha helices, so you have a bit more structured conformation, and it's reducing the local gyration radius. And then I just want to very quickly show you what we did also to search for binding pocket, because we were interested to that. So actually I'm comparing, so I have a parameter which is defining the pocketness of a residue, whether a residue is at a good position for pocketness. And if we are looking at the flexible mechano conformation, we have a very flat pocketness portedness, relatively flat portedness profile. But for the structure generated with TIBP, we have many residue which have a maximum of pocketedness and giving rise to this kind of pocket, and that could be good candidate for starting to do mutation to see whether we can have an influence of the interaction of SERV1R with alpha-synuclein, for example. And okay, so now I just presented you a geometric approach for systematic exploration of the conformational space for intrinsically disordered protein, and we can use a parallel different method for prediction, which are more or less related to, which are more or less devoted to more folded partially folded protein or completely disordered one. What we are obtaining, we are a similar distribution of coordinate and radius of gyration that conformation generated by another method, and we are able to actually to look at the pocketedness, give you some the TIBP conformation give you some peak of pocketedness which can be useful for searching for binding pocket. And here I just want to acknowledge my co-workers. Thank you very much for your attention.
Thank you, Teresa. Okay, we have time for a quick, fast question. No questions? Yes. Oh, well.
Is there any issue with clashes, atomic clashes? So whether the data of the protein database is reliable, or do you address this?
Okay, the database, okay, I was using the PDB, which database? About the folded protein, about the folded, these statistics here, you are talking about because actually when I'm doing this one was taken from the PDB and there is no clash. Here I'm doing IBP and I am spanning the tree, but I am preventing that there is no clash, because actually I am taking into account the van der Waals radii of the atoms. And then the place where you can get clash, actually you are getting some, but it's here when you are assembling the fragment you obtain for the conformation you obtain for the fragment. And here in magenta you have in red you have the number of clash, and you may have quite a lot of clash, especially but okay, that's can go up to 30% of the conformation you have to remove because you have some clash. Yeah, the clash are relatively important for reducing the conformal space. Yeah, you're right.
Okay, thank you very much, Teresa.